Just to, you have understood what is important to understand before going to the real possible application of the SNPs analysis is, first of all, you have to remember what a SNP is. It's a single nucleotide polymorphism, means a mutation of a single letter. So we are done by letter. Our DNA is a long sequence of letters. Now we have the access to the entire book, so we can look at the single genes and look very easily and incredibly also in a fast and cheap way. I mean, the technology available to detect the presence of a mutation in a person is extremely cheap and fast, and 5 years ago was not the case. Well, so, and important is to understand that the mutation, the change of a single letter in the sequence of a gene will produce a protein that not necessarily is alterated, sometimes is just lightly modified, sometimes change the ability to link the substrate if it is an enzyme, and the level and the range of these changes in activity can be linked to the presence of other polymorphisms. This is to let you understand the first sentence, the first claim that we did at the beginning. So nutrigenomic is a fantastic clue to identify a risk factor, but it's not a sentence. So at the moment it's impossible to look, it's not scientific to look at one gene or 10 genes and identify the real risk of a disease. You have the possibility to identify a predisposition to a disease, but it's just a difference in terms of percentage to having it or not. That is not a real sentence. So, as I told you before, I will jump many slides because otherwise the genetic part is very complicated, but it's important to understand that the SNPs approach is widely used by pharmaceutical company to try to create a sort of individualized therapy. I mean, the possibility to identify before the beginning of a therapy the subjects that are well or not responding to the therapy, or more importantly, look at this. This is quite impressive, but the 6th leading causes of death in hospital is adverse drug reaction, I mean, in the world. So the first one is cardiovascular disease, but the 6th is related to adverse reaction due to mutation that alter our ability to metabolize the drug. So a drug that is a perfect cure for one disease can become a real poison for another person with the same disease. And this is the goal of pharmacogenetics, that is exactly the same things of nutrigenetics, but doesn't look at the nutrients, look at the drug. So it's a little bit more financed because there are many companies interested in this field. This is just some example. There are many mutations known in the phase 1 metabolisms, the cytochrome P4050, the compounds that used to metabolize drugs. Drugs are xenobiotics, means substances that are not part of our biology, so that are aliens for our biochemistry. So when you assume a drug, this is important because many of the nutrients are the same. When you assume a drug, the first reaction of the body is to metabolize, means to modify the structure of the compounds in the attempt to destroy it, to clean the body from that compound. So the first barrier is done by these cytochromes, and there are many SNPs known, diffused between the population, that influence the ability of cytochromes to work. So this is not only very well known, but is also for some types of therapy also used in the clinical practice to look at the genetic makeup of a person before starting a therapy, to understand if it will be a good metabolizer of that drug. I will go a little bit. I'll come back on that. Okay. So our goal for this part of nutrigenomics is the possibility to identify mutation of genes that are linked to the ability of a person to utilize the food, or better, to identify some mutation that improve the possibility to get a disease, and identify a possible nutritional suggestion to interfere with that predisposition. That means to alter the bad predisposition utilizing food. So to get this, excuse me, I was I have the tail, so it's quite complicated. As I told you, the problem of having a real picture is related to the complexity of the gene-gene interaction and gene-environmental interaction. So when you have a single data, you have just a small spot, a small dot in a big sea. But from that point, you can start a rational approach to a personalized nutrition. We were discussing before with one of you, one of the biggest mistakes that was related to the advances of science, of nutritional science, is that till now a part of children and geriatric person, for a nutritionist, every person is similar to the other, so there is not individual difference in the ability to metabolize food. So the pyramid that we have seen at the beginning, the Mediterranean pyramid, the Mediterranean diet, by dogma is the same for everybody, and this is not completely correct. So what we have seen till now in this small presentation is that each individual is different from the other, not only in terms of physical aspect, but also in terms of the ability to metabolize food, drugs, and to interact with the environment. These are a small list of some SNPs that can interfere with some nutrients, that can be associated with the needs of some specific nutrient. Now we'll go deeply on the single nucleus. So, I told you before that SNPs, so the polymorphisms is the goal. So, from a scientific point of view, from the side of the research, the problem is to discover all the SNPs that are present in a population and to associate these SNPs with a specific phenotype, with a specific trait, with something that can be also the risk of a disease. To realize this, there is at the moment a lot of investment. There are two major consortiums dedicated to the SNPs research. One is in the States, the other one is in Europe. The European one got a lot of support by pharmaceutical companies because it's a little more dedicated to the pharmacogenetics. Some of these projects already produce website where you can have access to have a picture of the SNPs present in the world. Till now have been recognized 1,500,000 SNPs